We're moving on to our third panel, one that we're very excited about, as we all are, all of them. But this one, I think, invokes some of what Vince Bonham talked about this morning in terms of looking at who's coming up behind and how are we engaging with this next generation. The title of the panel is Insights from the Field, Next Generation Native Researchers. So this is our opportunity to talk with those who are choosing science careers, who are choosing to look at genomics and other kinds of research, and to hear some of their stories about what their journeys have been like, and to learn about some of the amazing, innovative science and culturally based approaches to work that they are engaging with. So we're super excited to have this group. And leading the way as our moderator, a good friend, colleague, Dr. Dorothy Aguilera Blackbear. We're very excited to have her with us. She is Choctaw and serves as the Vice President of Research and Sponsored Programs at the American Indian College Fund in Denver, Colorado. She earned her PhD in Educational Foundations, Policy and Practice at the University of Colorado at Boulder, and she served previously as the Director of Institutional Research at Northwest Indian College in Washington State. Any opportunity to engage with our tribal colleges and universities is very important to NCAI and our partners here. Dr. Aguilera Blackbear is committed to research training and leadership development amongst American Indian Alaska Native students and faculty. Please welcome Dr. Aguilera Blackbear.
Good afternoon. Wow, these lights are bright. We do have stars on the stage right now. These are truly the next generation of Native researchers, and I have had the opportunity and pleasure to be able to interact with them today. So thank you for that. I really appreciate it. So let me begin with introductions, and our first researcher is Keolu Fox, and he is Kanaka Maoli, which is Native Hawaiian, and a genome scientist in his fourth year at the University of Washington's Genome Science Doctorate Program. His research focuses on discovering genetic variants that contribute to health disparities in diverse populations. Mr. Fox currently works with Seattle's Puget Sound Blood Center, where he looks at the potential for next-generation sequence analysis to increase compatibility in blood transfusion therapy. He hopes to give back to his community of Polynesians so that they might also benefit from developments in genetic research. Beyond several student senate leadership roles in academia, Mr. Fox is interested in engaging with the science policy arena, addressing issues that will impact the future of genomics. Please welcome Keolu.
Just choose it. Where is it? Fox, there we go. Yeah, I think I just do where's the little, is it? Oh, okay. I'm a Mac user, so. Oh, from the beginning? Okay, right on. And that guy, bottom? Is it the bottom one? Bottom one, let's try. No. Okay, cool. All right, thank you. All right, thank you, everyone. Thanks for having me. This is a huge opportunity and greatly appreciated. Aloha kako, that means may there be love between us, but not that kind of love, right? And my name's Keolu Fox, and my Hawaiian name means the steadfast one, or the keeper of the faith. And I think we come from the Big Island of Hawaii, but I was back there in February and we had a conversation. And our family donated a kahili, which is a traditional flag, to, I believe, the Bishop Museum. And we were having an argument over whether it was black or purple, because they're pretty close. And if it was black, it would be from Queen Kaha'umanu, who is the queen and the favored wife of King Kamehameha I. But if it was purple, it would have been from Kauai. So this is a serious issue in that we don't know which island we're from. We really believe we're from the Big Island. So this is one way where we could use genomics to repatriate our identity. I don't feel like I'm a Native American. I feel like I'm Hawaiian. I feel like I'm Polynesian. But in that sense, we're all the same, right? So my research actually centers on utilizing next-generation sequencing technology. I'm in technology development. I work with hardware. I design algorithms. I work on the bench. I move fluids. We do it all. I work in science policy, and I'm at University of Washington. My boss, or my PI, is Debbie Nickerson. I specifically work on next-generation blood type sequencing. So we traditionally use serological methods to type blood, and we've been doing that for at least 100 years. And this is kind of an analog process where you observe clumping patterns. But what we do is we know the genome's digital, and we utilize that digital perspective to increase the resolution at which we type blood. And I'm going to tell you a little bit about that today, and I'm going to give you a tune-up on genome sequencing as well and where we're at. So first I want to talk a little bit about genomic economics. And here we have a plot of where the price of genome sequencing when the genome was first sequenced in 2001. You can see it's almost at $98 million to sequence the first draft of the human genome sequence. And these are the machines that we were using. They're called Sanger sequencers. They're pretty slow, pretty accurate. Now, when we moved to 2007, you can see the introduction of next-generation sequencing, and I'm not going to belabor that, but just know that the chemistry is more efficient, the hardware is more efficient, and the algorithms we use are more efficient as well. And you can see a picture of one of these, NGS. And this is kind of, in 2010 is where I entered the game. I've seen genome sequencing change in the last 4 years. There's been a revolution in medicine. I'm going to tell you about that today. By 2014, we have these benchtop sequencers, which are the size of a Keurig. You can find them, in any, hopefully in any hospital in the world in the future. And then you can see we have Moore's Law and just how cheap genome sequencing is becoming. And it's around $3,800 for a short-read genome sequence, okay? We always say from base pairs to bedsides. There's 4 different base pairs. We want to bring it to the bedside. We want genomic medicine, predictive and preventative medicine, not reactive medicine. And so you can see that we have the human genome as a reference. So every time we sequence a reference, we compare it to the human genome reference, which was sequenced in 2001. It's become more accurate. The second concept that I alluded to is this idea of the digital genome. This is digital information. These are bits of data. A genome is 6 gigs of information. We have to store it. We have to manipulate it. We can write algorithms to do many different things, okay? I want to talk about a little story about this kid named Nicholas. Nicholas was 3 years old. He had a serious stomach disease. No one understood what was going on at all. Here you can see him. He's got the coloscopy bag in. He does not look happy. How many rounds of chemo, right? Enter human genome sequencing. There he is. The true hero of this story is Nicholas. But his Robin is next-generation genome sequencing. We were able to identify a specific genetic variant in a type of sequencing called exome sequencing. And I'm going to tell you a little bit about that today. So here is the way that normal stretches of the genome are composed. And you can see each genome, give or take, is 3 billion base pairs. It's actually 6 billion because you have 2 copies of each gene. And it's composed of components called introns and exons. But we know that 85% of the heritable disease in human genome is found in the exon, right? So 2,200 exons about in the human genome. And that's about 30 million total base pairs, or 1% of the genome. And if we were to look at a little comparison with an apple, it'd just be one little slice, right? But how do we capture just that 1%? Why would we inundate a computer server or a clinician with all of that information if they only need that 1% that actually codes for proteins? Well, what we do is we start with that DNA, then we perform something that's called a library prep, and we turn it into a shotgun library where we use different methods like chemical methods or sonication with sound, and we chop up the genome into these tiny pieces. And here in blue, you can see that those are likely exonic sequence. Then we hybridize them to biotintillated probes. And what I mean by hybridize is that Because the genome is reverse complementary, right? 5' to 3', 3' to 5', you can attach probes and program probes so that they only capture that exonic sequence. Then we use a bead cleanup. We attach them, right? And then we elute, wash, voila. All you have left is the captured exome data, and then we shotgun sequence it on a next generation sequencer. So we can use this application for a lot of things. Undiagnosed disease like Nicholas, the number 1 and number 2 killers in the United States of America, heart disease, cardiovascular disease, right, and cancer. And then pharmacogenomics, which you heard about a little bit earlier today. But today I'm just gonna talk about blood typing, traditional blood typing. So you can see I have a little picture of these red blood cells, right? So usually when I give this talk, I always poll the audience and ask them, what's your blood type? But I just recently saw some statistics that more people know their horoscope than their actual blood type. Which is god-awful, right? Because your blood type is gonna contribute, it's a risk factor for how you're gonna die. I would say that it's really important to know your A, B, and O blood type. And then I wanna make the argument that it's even more important that we should know subtypes within A, B, and O. And we're just gonna talk about ABO today because it's the most medically relevant blood system, and we've probably all heard of it. If you haven't, you should probably figure it out. So here I have a map of all the different blood systems. And you can see there's 34 known blood group systems. There's 339 serologic phenotypes. These are clumping or aggregation patterns, and there's 48 known genes with around 1,100 alleles, which are specific genetic mutations. And then I have the year discovered. You can see ABO down here at the bottom. It's a canonical carbohydrate. I'm not gonna belabor the biochemistry. You can ask me questions later if you like. And there are 2 antigens, right? A and B. So the other idea is, why does blood type matter? Why is it significant medically? It's very significant because you should think about blood as a temporary organ. Each unit of blood lasts around 40 to 120 days. So blood transfusion reactions occur all the time, especially people who receive multiple transfusions. That's like an individual who has sickle cell anemia needs a transfusion a month. Cancer patients who receive chemo are repetitively receiving blood transfusions. And then we have mother-child incompatibility, such as hemolytic disease of the newborn, right? So as I alluded to earlier, these ABO blood types are linked with disease. And you can see in red, I have the myocardial infarction or heart attack is associated with the A or B or AB blood type. VTE or venous thrombosis is associated with the A or B blood type, and then coronary artery disease as well. And then specific types of cancer are associated with O. It's also important to know that the inheritance of blood group alleles works like this. You receive one from your mother and one from your father. In this case, this person's AO, A on one gene and O on another, because you have 2 genes. You have 2 copies of the ABO gene, right? This person's BO. These are all the possibilities for their children. You can see that O is recessive, and you can even end up with the AB blood type, which is really unique in the human genome. It's called codominance. It's the classic example in a genetics classroom of codominance, right? Okay. But the way we type blood today, we use agglutination. And there's a fundamental idea in immunology that there's self and not self. Our body needs to be able, our immune system needs to identify things that are not within our genome. And that's exactly what these serologic tests do. And you can see that if you're A blood type, you wouldn't have anti-A antibodies. And so this person's A positive, and D is just another antigen group system. So just to further that little genetic tune-up, I want to bring you guys back to that idea of the reference. And here we have the ABO gene. It's composed of 20,000 base pair, and then the exonic portion is only 1,000 coding, right? And that's about proportionate to that apple I showed you earlier. We have this reference model, and there's 7 exons total, right? So let's take a look at the first motif or number of variants that associate together on a background. And in genomics, we just call that a haplotype, right? You've heard people talk about MT haplotypes earlier. And you can see it's CG, CG, and that's associated with the A haplotype, right? And this, whoops. Oh, getting a little, here we go. Okay, in this situation, you have the B haplotype, and you can see there are just differences in those genotypes, those specific genetic variants here. Then, in the situation of O, we actually have this frame shift or deletion, and it causes the degradation of this portion of the gene. In this situation for A2, you have a frame shift, which causes the addition of 21 amino acids, which is clinically significant. And that's back to this idea of subtypes. So antigen density, which is, you can see the density of these antigen carbohydrate structures on an A1 individual versus someone who's an A2 individual, is very important because it differentiates the way that these clumping patterns occur. Additionally, you can see that A1 versus A2 organ transplant success rate is extremely important. You cannot give somebody who's an A2 an A1 organ because it could potentially cause antibodies and they might attack that organ. Another thing that's really interesting is that population frequencies of these various haplotypes vary per population. So European individuals have a higher frequency of having this A2 subtype. So just briefly, this is the most hardcore data slide I'm going to go through today. And I designed an algorithm to predict not just blood type, but subtype based on exome sequence. So here's how it kind of works. I showed you those haplotype motifs. You can see them. There's A1, B, and O. It's all derived from exome sequence data. Then we have these ABO subtype calls from 160 chromosomes. That's 80 people total, right? And you can see it on the subtype level. The most common haplotypes are O haplotypes. It's just like anything else. It's much easier to break something with a frame shift than it is to accumulate variations that would result in a gain of function, right? Then we pair them and we impute the blood type, and then we compare them with the actual serological data, and you can see that they're 100% concordant. So I was really stoked about that. One thing we can also do with exome sequencing is because we get such a high resolution look at the genome, we can discover new blood types that might have clinical significance. And here I've just provided you with a stick model of that gene. Here's all 7 exons, 354 amino acid residues in total. And then I have in green the ones we've discovered using exome sequencing. Here's our traditional frame shift deletion, the loss of function deletion I discussed earlier. And then we have insertions and deletions that we've discovered and known variants that have RSIDs. That's our way of cataloging known human genetic variation, right? So it's pretty cool because we can actually discover clinically significant blood types and subtypes using genome sequence data, which is new. So in summary, I told you about a computer program that I've used to type blood and subtype blood. Additionally, I told you that our method is extremely accurate. It's just as accurate as the clinical golden standard. It's higher resolution than any antibody test that's available. We can look at extended haplotypes, or what we call phase, and we can differentiate between someone who's AA versus someone who's AO. And we can discover new blood types or subtypes. I've discovered a weak B subtype that I didn't really discuss that much today. And then understanding the clinical importance of transfusion therapy. By identifying all of these rare blood types in rare populations, we can increase the resolution at which we cross-match blood in the clinic, and that's really important for transfusion and transplantation therapy. So in the future, what do we want to do? Well, we want to continue investigation of all of these rare alleles, all those things that we've discovered that we didn't know existed prior to using next-generation sequencing technology. And we can associate those with actual blood types by associating genotype to phenotype, bringing it around, bringing it back. And then apply this more broadly to other blood group systems. I told you about ABO, but there are 36 other ones, right? And there are subgroups within those and so on, in various shades of variation that I didn't even talk about today. Finally, I want to talk about an idea that I was very excited to talk about it. And it started with a journal club that myself, Katrina Claw, and Joe Yurchet at the University of Washington would have at lunch, where we would read human genetic variation sequencing papers, and we just noticed that there weren't very many that focused on minority populations. And that's what this plot shows. shows that 96% of genome-wide association studies have focused on European populations. 4% have focused on non-European populations. There was a panel called the 1,000 Genome Projects, and they included 1,000 different genomes. Guess how many of them were Hawaiian? Anyone? Yeah, big old goose egg, none of them. So our whole idea and crusade is to increase the populations that we sequence. DNA sequencing of 10,000 people we've realized that rare variation is population specific and it's associated with disease. And we can do all kinds of fun things for migration patterns like you heard about earlier. It's really important to consider that in medical sequencing in all populations. And I just think it's really important for all of us to participate at all levels. At all levels. And I mean from top to bottom, from your high school teacher to, a lot of people are afraid to learn how to code. A lot of people don't want to learn how to move fluids around at work. Everything. Learn how to write grants, learn how to write papers. It has to be for the people, by the people. All right. I would like to thank Debbie Nickerson for giving me the opportunity to work on all this wonderful data. I'd like to thank Ian Stanaway for helping me learn how to code. Jill Johnson at the Blood Center for giving me access to a lot of serological data and perspectives, clinical perspectives. Chen Yi, David Crossland for helping me out with a lot of my statistical methods and just raw mathematics. My whole lab, the committee members that have made me a strong scientist, and my whole family, my ohana right here. Mommy, aunties, my dad, and my beautiful girlfriend. So I'd like to thank the NCAI. I'd like to thank the Smithsonian. It's a dream. You always hear about this place. I can't imagine, talking here. UW School of Medicine. Yeah, Genome Sciences. NHGRI. NHLBI for my funding my PhD. And the Puget Sound Blood Center. And don't forget to donate. Thanks, guys.